It's still going. There we go. And then we can hit record.
I think it's going to be a short talk anyway. I've scheduled 2 hours, but it's never going to take 2 hours.
Okay. Right, there we go. Hello, everyone. Welcome to BGA24's 18th session on Wise Ancestors with Celine Corriard. Please take it away.
So hi, everyone. I'm going to share my screen. Okay, hopefully it's as the right mode. So today I'm going to talk to you about wise ancestors, and I'm very excited about presenting all of that to you. Just first, a few words about me, just so that you know who you're speaking with. I'm Solène. I am currently talking to you from France. I obtained my PhD in molecular biology and bioinformatics in France, and then I did 2 postdocs in Canada, one on inequities in human genetic care, and one in biodiversity genomics, working on the Canadian Biogenome Project, during which I've met a few of the people I see attending here. And now I am the scientific director for Wise Ancestors, and I'm going to introduce Wise Ancestors in a couple of slides. I also wanted to mention that I am a Nextflow ambassador, and last year during BGA23, I did a session on introduction to Nextflow. So if you want to discuss Nextflow for biodiversity genomics, I'm also happy to discuss that. And I am a member of the DSI Scientific Network, and I'll be working with the network next week during COP16, COP Biodiversity in Cali, Colombia. And so just about the workshop structure, there is a first part that's going to be end off, so it's a bit of a presentation, but there isn't too many slides about Wise Ancestors model. What is Wise Ancestor? What is our mission? How does it work? And then the more hands-on part, it's, as Damon mentioned, about our platform. So an overview of the platform, what do we call conservation challenge, and also examples of them. And then you'll be able to register as a collaborator on the platform if you want. And so what is Wise Ancestors? So Wise Ancestors is a lot of things. It's a nonprofit, so we are not academia, we are not private, we're a nonprofit. It's still relatively new. I joined them less than a year ago, but before that, it's less than 2 years old. It's both a model and a platform, which I'm going to explain in a second. And it's collaborative. And that's why we need you. And it's also an EBP affiliated project. I think most of the people in the room are aware about what EBP is. So EBP is the Earth Biogenome Project Consortium, and it regroups a lot of projects who, among other things, generate reference genomes for species. And so we could wonder why an EBP affiliated project is presenting at BGA24, because I think we're the only one. And BGA is about how to use bioinformatic tool that underpins the EBP. And the reason for that is because Wise Ancestor has a decentralized approach, which means we don't have labs ourselves, whether wet labs or dry labs. We don't have servers. We rely on other labs, platform, and scientists to participate with us to do the work. And so I'm going to present to you how we do that in a second. But that's where we need you, because it's really a collaborative approach that is going to really fit in the other Earth Biogenome project to meet the collaborative points of sequencing Earth biodiversity. And so Wise Ancestors' mission is spreading biotechnology, indigenous science, and traditional ecological knowledge to conserve biodiversity. And each word in this mission really means something to us and was really thought. And so I'm going to explain why each word is so important using the 2022 Kunming-Montreal Global Biodiversity Framework as an example. So some of you may be aware of the GBF, but in the Global Biodiversity Framework, which is accessible online, they have 23 action-oriented global targets for urgent action to conserve biodiversity. And target 4, which is probably the one most of people working on EBP are aware of, is halt species extension, protect genetic diversity, and manage human-wildlife conflict. And within the target, they use a lot of biotechnology tool to conserve biodiversity. So why we have biotechnology to conserve biodiversity. But we believe that indigenous science and traditional ecological knowledge are also essential to conserve biodiversity. In 7 of the 23 targets, so 30%, indigenous people and local communities are mentioned, including target 13, when they say increase the sharing of benefits from genetic resources, digital sequence information, and traditional knowledge. And we really see biotechnology, indigenous science, and traditional ecological knowledge as 3 entities that can thrive on their own. And we want to see how powerful they can be when bred together respectfully to conserve biodiversity, and really bringing together those knowledge, those different type of sciences to see how we can work together. And that's really Wise Ancestors' mission, and I'm going to show you how we want to do it in a second. And we also say breeding, and breeding really means that the 3 entities, as I said, can thrive on their own, and none is going to take over the other. We really want to have that respect for each other that hasn't necessarily been respected in the past, and recognize that there may be boundaries, and it's not about merging or really taking over. It's really about, with the boundaries that may be needed, seeing how we can work together. And so I would like to mention target 20 of the Kunming-Montreal Global Biodiversity Framework, which means strengthen capacity building, technology transfer, and scientific and technical cooperation for biodiversity. And so that's really what we are about. And we really think it fits within the EBP as yet another project that will make it possible to sequence biodiversity on Earth. And so, depending on where you're from, you may be more or less aware about indigenous people and local communities linked to biodiversity and involvement with biodiversity. But it's important to know that indigenous people and local communities are key stakeholders of biodiversity. It's been recognized by the United Nations and by many, many groups now. They successfully stewarded their lands for millennia. They have traditional knowledge that provide additional information about the species and the environment and the ecosystem that was known by Western scientists. And that's also something we really want to reflect through our different conservation challenges, that the ones I'm going to present to you today, they could not be done without traditional knowledge. Like if we were scientists, so today I'm going to present challenges that we're co-developing with Bermuda. With our knowledge, if we were to go to Bermuda, we wouldn't be able to do those conservation challenges. And that's what I think is really important, is to recognize that it's really bringing together different sciences that can lead to those projects. And the other thing I'd like to mention is that the social political reality is that biodiversity and genomic resources are not evenly distributed on Earth. Genome sequencing and biobanking requires expensive equipment, laboratory access, skilled workforce, and computational resources, which is not always available to indigenous people and local communities. And so one of the idea of Wise Ancestors is to create those connections, to create those collaborations, so that those fact that it's not evenly distributed can be sort of tuned down, and that access is improved for everyone. And so how do we want to do that? That's big words. That's nice to say. Everybody kind of says that. So we came up with a model. And the model starts here with outreach and relationship building. So for people who have done it in the past, they know that it takes a lot of time. So it's just one sentence and one part of the model I'm going to present, but it's probably the longest one. We're reaching out to communities or to local scientists that we may know of or that we find have a certain connection to the land, and we present to them our model. So pretty much what I'm presenting to you today, we would present it to community members who are engaged or to local scientists, and we really just start the discussion by presenting the model, presenting what we can do, and see if they want to work with us. And so if there is an interest, then we start a step that is the co-development of the project. So our project are called conservation challenges. And so it's really a co-development because the project will come from a discussion. It's not us suggesting a species, it's really a discussion about what's needed to improve conservation on their land, what species needs to be studied, whether it's an endangered species, an invasive species, a food source, anything. So together we identify the species of interest. We define the technical work that is needed. And what I mean by technical is biotechnology. So would sequencing help? Would population genomics help? Would biobanking help? Like, what could Western science bring to help conservation on the ground? Each conservation challenge is also associated with a benefit sharing component. So the benefit sharing component is still, it's not very defined, but it's something we discuss with the community as we found a project that is going to help conservation on the ground. So I'll give 2 examples later, but it can be land stewardship program, so that they can acquire land and replant native plants or work on with their traditional knowledge to restore the ecosystem. Can be capacity building. It can be knowledge transfer from elders or knowledge bearers to younger generation. So anything that they can see would help conserve their species on the ground. And once all of that is defined, we work to obtain consent and build mutually agreed terms. Again, consent can take different forms. We like paperwork because we're Western, but we understand that it's not always the way to go. So we discuss to see what form this should take and what should be included in the mutually agreed terms. So that's sort of the invisible part, the community engagement part. It's what's taking the longest. And then the second part is the publication of those conservation challenges on the Wise Ancestor platform. So once we've came up with all that, we published the conservation challenge on the platform that I'm going to show you in a minute. And that we use this platform to first raise funds to pay for those challenges. So we do crowdfunding, but we will also apply to specific grants. For example, if we work on a certain country, we will see if there is grants in that country that we could apply for to fund the challenge. We have different ways of fundraising for the challenges. And then on the platform, and this is where you come in, we recruit collaborators. So each project, for example, generating a reference genome, will be cut in different steps, such as sampling, sample processing, sequencing, bioinformatics, and we will look for collaborators who can do those different steps. and we will create those collaboration. We will be responsible for making sure everything goes smoothly. For example, the analysis will happen after the sequencing. So once the sequencing is done, we'll connect with people doing the analysis, etc. We will generate the final reports, which will merge the results from all the different scientists who work on a given conservation challenge. We will compensate, or we'll pay the collaborators for their work. And the most important is that we will return the results to the community. And that has not been always done in the past. And that's why it's very important for us. And we really mean it. We want to return the results as actionable results. Like, well, of course, we will return the reference genome, but we want to tell them what it means to have a reference genome and what they can do with it. And we made it a circle because we really hope that the communities will have enjoyed the process, seen a benefit from it, and will want to go again and work on a different species or in a different environment with us and do this all over again. Is there any question or any comment?
So this kind of acts like a water wheel, in a sense, then, doesn't it? So as you work with the community and eventually they're given their results back, it should help build local knowledge in a more scientific framework to help them further go on and do it themselves as well.
Yes, if they want to. Some communities may not have the bandwidth to have increased capacity in biobanking or sequencing, for example. That's why the benefit sharing part is really open for discussion, and it's really what would benefit them. If it's knowledge transfer and they want to learn about sequencing or biobanking, then yeah, of course, and they will then probably the younger generation will take over. If it's not something that they can prioritize at the moment or that they want to do, then but yeah, there's definitely going to be, even during the outreach and relationship building, we can see that there's a lot of targeted question that comes after 1 or 2 meetings, because there is an increased understanding of what biotechnology can do very quickly. That a lot of people have worked, including people in this call, worked on creating resources that are accessible to explain, like, what can we do with a reference genome. So I think it's really easy to understand. See something in the chat. How long does the community engagement take usually? Are you working with different communities for different species? How long does the community engagement take? We don't yet have enough, so I'm going to present 2 challenges today, and we're going to launch 5 more in the coming days, but those were developed in 2 different communities, so we don't have enough experience to really give something like real number, but I would say up to a year. And that's why it may not be feasible within academia. Like, I've worked in academia and I understand the system, and some people are able to do it within academia, and that's very impressive. But with the grant cycles and things, it can be challenging in academia. Here we have different challenges, so we can take that year to do it properly. But yeah, it takes a long time. And are you working with different communities for different species? Yes, I'll present in a couple of slides. So we've worked with community in Bermuda to work on a specific tree, and we're working on a different country on bird species, and we hope that we'll be working on other species soon. I cannot moderate the Discord, so Damon, if there is any question on the Discord, please let me know. Thanks. Great. Okay. Okay, so questions. I don't know what time is that. I was thinking maybe we could take a break, but we'll keep going and we'll maybe fit it in an hour. I don't think people need a break after 25 minutes. So now we'll get more into the platform. But again, if you have any question about the model at any time, just unmute yourself and ask or put it in the chat. Okay. So this is the platform. This is what it actually looks like. So we have a website where all the information about the nonprofit is. And then we have the platform, and the platform is really more for donators and scientists who wants to collaborate on the different challenges. And so we will soon publish challenges that are happening in Colombia. We will probably launch them during COP because some of our team will be in Colombia for COP16 next week. So stay tuned. You can go back on the platform in a few days and see them. And the two challenges that I'm going to present to you today are challenges that were co-developed with local scientists in Bermuda, working on a tree called the Bermuda cedar. It's an endemic tree of Bermuda that is critically endangered. And from one species, we broke it down into two challenges. The first one is to generate a reference genome and an annotation for that tree. So there is not yet a reference genome for this tree. So we'll be working with the local scientists to do that together. It will be included in the EBP metrics, like it will meet the EBP standards. And the second challenge is the development of a genetic test to detect hybridization of juniperous trees and then biobanking. And so I'm going to explain that one in a bit, but you'll really understand how from a reference genome, we can apply this knowledge to help them on the ground in their conservation work. And so, yeah, big release. This is the link to our platform. We're launching it today, so it's finally here. You can scan the QR code, or you can access the link. I'll put it in the chat. And I invite you to go on the platform. So, oops. So yes, this is the platform. You can see that I'm not signed in. We only have, as I mentioned, 2 real conservation challenge right now. This is more of a teaser for donors or for anyone who would feel connected to one of those challenges or who just want to donate. It's possible to fund the challenges. So by clicking on this button here, sorry, apparently my computer is slow this morning. You get on a page where you enter your personal information, how much you want to give for that specific project, and then you have your payment information. It goes through a secured interface, and that allows us to crowdfund for different challenges. Donations are tax deductible mostly in the US to the extent allowed by law. So as I mentioned, crowdfunding is one of the funding methods for our challenges, but you can see that we've already raised a lot of money for those challenges, and those are mostly through grants application. And so, yeah, we will really look into different ways to fund the challenges. And then if you click on plus information, for each challenge, you get more information. So you get the species name, the scientific name for the species, an overview of the project. So as I said, this one is about Juniperus bermudiana, so the Bermuda cedar. It was one of the most dominant trees in Bermuda, and it's central to its history. And we aim to generate an annotated reference genome to support conservation of this critically endangered tree. And so for for each challenge, you can have more information, such as the short introduction, goal, methods, and the expected impact. The method here is very interesting because in Bermuda, they have a nanopore sequencing machine, one of the latest one. And so we're working with them to keep all the work local. And so all the sequencing will be done on nanopore, which means long read, POR-C, and direct RNA sequencing. And it will be one of the largest genome assembled and annotated using only nanopore technology. So we're also partnering with org.1 on this one, which is nanopore branch for biodiversity. And we're very excited because this one is also a bit of a technical challenge as much as a conservation challenge. And also for each challenge, you have the benefit sharing associated with this project. So as I mentioned, for this project, we've partnered with local scientists in Bermuda, working for BioQuest, which is another nonprofit. And we've worked with them to define what could be the benefit sharing. And so we're partnering with Bermuda Department of Culture to support a program that already exists. It's called the Cultural Apprenticeship Program, or CAP, which pairs a tradition bearer with an apprentice. And so we will do a CAP, especially on Bermuda setter. And so a young Bermudian will partner with different knowledge bearer across the island to learn about the botany, the ecology, the propagation of that tree, to really make sure that there is a connection and to support the work they do on the ground. And so, yeah, we're pretty excited about that one. It really came together nicely with the Department of Culture and BioQuest and us. So, yeah, that's going to be a nice one. And below, you can see what we call the phases. So each conservation challenge or each project is broke down into different phases, so different steps. And so those are pretty usual. So there is a sampling step, of course. Then there is sample processing or sample preparation. So there is a certain wet lab experiment to do to prepare from the plant to the library before sequencing, cross-linking for POR-C or RNA extraction for direct RNA sequencing. Then there is 3 steps of sequencing. We sequence using long reads, but we will also be doing POR-C and direct RNA sequencing. There is some data storage that is required before processing the data to make it so. But we will store the data locally to process them, generate the reference genome before releasing them on ENA or another INSDC. And then there is the bioinformatics step, where the genome, the assembly and the annotation will be generated, and also the benefit sharing component. And so for each step, I'm going to take the bioinformatic analysis as an example. But for each space, you can click on it and get more information. So as I mentioned, for example, this one is very exciting because it will be, to our knowledge, one of the largest genome to date assembled using only nanopore data. And so we are looking, it says ready for application, because we are looking for scientists, bioinformaticians, who, if we provide them with long-read data, POR-C and direct RNA data, could generate the reference genome and the annotation for this species. And so partner with us and with the local scientists to make it happen. We do not expect people to do it for free. There are dedicated funds for each phase. We do hope that in some cases, some labs or some team will ask for less than the dedicated fund For example, if they already have servers and already have a pipeline ready, then it's probably not going to cost them that amount of money. But that's really the idea of putting our needs out there and hoping that the community will step in and say, yes, at the moment, I have capacity to do it. It will take me that amount of money. But we also understand and we know how things work, that some labs at some moment don't have the capacity because it's grant funding moment application, or it's, I don't know, some moments are just more busy than others. And so it's really about putting our needs out there. And if you have capacity at that moment when we need it, say so. And if you don't, like, we totally understand. And so for each phase, there is more data available. So for example, for the bioinformatic part, it's very basic information, such as what data would be available as entry data, so long read Por-C and RNA-seq, what type of analysis we expect. So we expect a de novo genome assembly and a genome annotation, what type of output we expect. So we expect a FASTA report as well as an annotation file. And so, yeah, this is it. It's also open for feedback. So if you have any suggestions for people who are from wet labs, I don't know everyone on the call, feel free to look at the sample processing information. For example, this one has a certain amount of dedicated found. We expect high molecular weight DNA starting from plant tissue, especially from young leaves. We suggested kit or a protocol, but we're open for other suggestions. And we expect some specific quality control, such as electrophoric, like an agarose gel, and the usual ratio to know the quality of the extraction, etc. So is there any question on kind of the overview of a challenge, or this challenge in particular?
I have one, if that's okay.
Yeah.
For starters, I think this is amazing. Like, genuinely, really, really cool. And my question is that, so anyone can apply for these positions then, or are you expecting there to be organizations that apply, or like essentially freelance bioinformaticians? And if so, how will you be going about vetting them? So obviously, it's all well and good having this great freelance bioinformatician, say me, for instance, but at the same time, because I've got no organization backing, how would you be able to verify that, yes, I can actually do the job?
That's a very good question, and that's a perfect transition to what I'm going to present next. So I can see that my camera is acting up a little. I'm sorry about that. I hope it's going to hold for the rest of the meeting. So if you're interested, or even if you don't think that a current phase or challenge fits with your capacity, you can apply to become a collaborator. And that's what I invite you all to do right now. So you enter your email address, and I'm going to answer to your question at the end of this little process. Put your real email address. I already have an account, so I have to put a fake one in, and I will delete my account. But if you want to register, please do. So you put your email address, you get an email in your mailbox that you have to approve your email address. So you're probably going to see my very messy mailbox. Okay, so I've entered my email address. This is just a step to verify that people don't enter just fake email address. So I can create an account. So you choose your password. You have to review your terms of use. I invite you to do it after the meeting. You can always delete your account if you want to do those steps at the same time as me. And if later you realize you don't approve our terms of use, first reach out to us because we would like to know. But they're pretty standard, and we expect that they're fine with most people. And then we ask some general information, such as pronouns. So again, feel free to do it at the same time if you want to apply for an account. So in the collaborator type, you see that we've came up with that idea that private individual can apply, and that's fine because I've been sort of in the same situation. And I also expect that some people may not, unfortunately, be supported by their institution to participate in it, but may still have the will to do so. So we understand that, and it's okay. You can apply as a private individual, again, organization if applicable. The contact email can be different from your registration email. We know some people have different mailbox, so that's fine. And then we ask, tell us the way in which your organization, and now I should say, or you, engage with Indigenous people and local communities in your work, and or how you acknowledge Indigenous rights and interests with respect to genomic and biological resources. So that's one of the first. It's okay if you don't do, but you have the will to do, and you're learning, and you're in the process of doing that. But as you were asking how we vet people, well, that's one of the way we want to make sure that people are not just here for the money, but also understand the whole model and the concept. We also acknowledge that people can just lie because that's free text. But we really want to understand why people joined in this platform, and if they're already partnering with Indigenous people and local communities, and if they're not, are they willing to put in the work? There are a lot of resources out there to understand all of that. So and again, that can take some time to write up. So you can put something like a placeholder at the moment if you want to keep doing that and come back later. Then you can save. And then the additional information is more about what you or your organization has the capacity to do. For example, if you're a bioinformatician, you will say, I have bioinformatic analysis capacity, and I can create a de novo genome assembly. I've done population genomic analysis in the past, or I've never done population analysis in the past, but I've done annotation, for example. But if you're more of an organization, like a big platform that apply, you may have bioinformatic and sequencing capacity and wet lab capacity. So it's really, this part is really about what capacity you can bring, whether as an organization or as an individual, so that when new challenges are posted and we have a specific need, we can send you a targeted email, be like, hey, we have this need. We see that it fits with what you've said you have as a capacity. Would you be interested in applying for this specific one? And again, like, we understand if it's not the right time, but maybe it's actually the right time, and you're like, yeah, good fit, technically good fit, timing wise. Let's work together. So this one is really easy to change on your profile. Then you can update it. For example, we ask if platform have certain sequencing capacity. For example, they may have an Illumina and a PacBio, and a year later get a nanopore sequencing machine, so they can just add and remove information very easily. We also ask, and that goes also with your question, if so that's mostly for big platforms if they have certifications. And that's one way of knowing if they are reliable, but that's mostly for wet lab and sequencing. I don't think there are much certification for bioinformatics at the moment. Maybe in data storage for security and tracking, but probably not in the analysis. So this is all information we gather. It's stored securely. And so you can save and apply. Sorry, I'm just going to say I have bioinformatic capacity. And I know how to do de novo genome assembly. I know how to do an annotation. And I apply. And so you can see that the layout didn't change. You still have access to the information of each challenge. But now you have a workspace. And you can see that it's waiting for approval. So at the moment, we have a step where we approve each collaborator application. So an admin administrator from Wise Ancestors review your application. We sort of expect to know pretty much everyone at the beginning because it's a small world, biodiversity genomics. We know the big platforms, and if you're a freelancer, someone probably knows you. We may reach out to a few people just to say, hey, like, do you want to have a 15-20-minute chat just so we know how you've learned about us? But that's more something because we're launching. We may remove that step in the future. So if you all created an account, you're probably in the waiting for approval state. I'll review your application later today. And you can see that you can reset your password or delete your account. If you just created an account, but you're not willing yet to know if you want to be a collaborator or not, that's fine. And you can see that you have no challenge at the moment. And so if we go back to the challenges, so I go back to the reference genome one. And if I go back to the bioinformatic analysis, now we can see that there's a button that says apply. You cannot yet click on it because we haven't approved your account. But once your account is approved, you can apply for a specific phase. And then we'll ask you, it's actually, I should have pre-approved my account so that you could see it. But we ask you if you've done that type of work before. And for example, if you have a publication or if you have a GitHub page with a similar pipeline. So those are the kind of information we ask to know if we think you're able to do the work. We also ask how much you are expecting to do that work, because as I said, we have a dedicated amount, but we hope that some people may say, well, it's only going to take me 3 days of work. I already have the pipeline. I have the servers anyway, so it's not going to cost me that much. So once we've gathered different applications for our one phase, we will use a sum of information to decide which collaborator is assigned the phase. And of course, if it's a collaborator that has done a lot of publication and their pipeline is publicly available and reproducible on GitHub, we will maybe go more for that person, that a freelancer that we've never heard of. But if the freelancer asks for less money, then that's also something we may need to consider depending on the grants we got. So there are a lot of things that we'll have to use to decide. It's really open to everyone to apply. We may run some interviews sometimes. We also want so each project is co-developed with local scientists and community members. So sometimes they also may want to meet the people who are gonna work on their samples or on their data. So we'll really play by ear. We don't have the full answer at the moment. Does that answer your question?
Perfectly so, yeah.
And so is there any other question about the creating an account or the application process? Do I have 10 accounts because I see that there's 10 people in the room, so do I have 10 accounts to approve later? And so I'll just use a bit more time. If there is no question, you can also take your time to ask the question, to talk to you about the second challenge, because I think it's a very, very cool one. So what happened with that tree, so the Bermuda cedar or Juniperus bermudiana, is that between 1946 and 1953, about 95% of the trees were killed by an accidentally introduced insect. And that led to massive deforestation. And those trees are very important for the island ecosystem because they cut down the wind, because they're like it's an island, so there is a lot of shore, and they cut the wind, etc. And so to respond, they introduced similar trees, so other juniperous trees, to protect the island ecosystem. And the problem with those, the intention was really good, is that apparently they don't have the same root system, and they're not as strong as the endemic Juniperus bermudiana. And so the local people are working hard to reintroduce Bermuda cedar, or true Bermuda cedar, the way they call them, in their ecosystem, so that they have strong trees that can hold the big wind that can happen on the island. But the thing is, the trees that were imported hybridized with the endemic Juniperus bermudiana, and they look very similar. So visually, they cannot make the difference between, is that tree more an imported tree, or is that tree more a Juniperus bermudiana? And so the goal of this challenge is to develop a genetic test so that they can, from a small sample of leaves, so very early when the tree is just growing up, know if that one is more of an hybrid or if it's more of a true Bermuda cedar. And so if and when we develop that test, we can see that will really lead to application on the ground, because they have growing trees at the moment in tree houses, and it's a need from the community to have that test and to very early know which trees or they should focus on and where they should plant them. And it's really, that project was born from that community engagement step. Like, we could have gone there and tell them, you don't have a reference genome for Juniperus bermudiana, we'll do one. But that wouldn't have probably been done. And from the discussion, we understood that there was an actual need, and that it can be done by really breeding our knowledge, because to create that test, we'll need to do a population study. So we need true Bermuda cedar and hybridized tree to sequence them and compare them. And that's known by knowledge bearers who've been on the island forever and who know which trees are what. Even though they look similar, they know from histories which one were imported and which one are not. And so it's really, I think this challenge is super cool because it brings together different knowledge. It will also be challenging technically because the test will be run using adaptive sampling technology from nanopore. And the adaptive sampling technology has been used mostly in human health, for example, cancer detection and rare disease detection, but it hasn't been used much yet in biodiversity conservation. So it will be about using also, like, technically it's super interesting to do that, and it will have a real impact on the ground, and they'll really be able to use it right away. And so for the benefit sharing of this project, it's the same as the benefit sharing for the reference genome project. So it's also the cultural apprenticeship program. because both projects are very linked. And so for this one, there is also sampling steps. So sampling of Bermudian cedar, but also sampling of the imported tree. DNA extraction for the different samples that will be done. Sequencing, again, some data storage, bioinformatic analysis. Then after that, once they're able to know which one are true Bermuda cedar, they want to biobank them, because they biobank juniperous trees growing in Bermuda, but they're actually not sure if what they've biobanked are endemic trees or hybrids, because the biobanking has been done after the introduction of the trees. So it's just a lot of very cool impacts on the ground thing that will happen. And that's really thanks to a collaboration and the group of local scientists and local knowledge bearer came together and agreed to talk with us. So we're very excited about that. And I hope you all are too. And I think that's about it. So I'll stop sharing. Again, if you have any question, you can ask now. You can also ask later on Discord. Oh no, I think I have a few more slides, sorry. That was me very excited about the launch, showing you the platform, seeing that there was no bug when I showed it to you. I forgot I had a few more slides. Yeah, so why should you become a collaborator now? Maybe you don't see a challenge that fits for you. We'd still like you to register the collaborator if you think our model is great. First, you'll be able to receive targeted notification when a conservation challenge that fits your expertise are posted. It will indicate also to us what expertise we have and so what expertise we lack on the platform, and we may target specific groups such as biobankers or others groups that we may need on the platform. And it will also help us grow a community, which will be very helpful to convince funders that our model can hold. So please join. You're the first cohort of platform testers, like literally, we just put the link on this morning. So we'll welcome feedback if it's technical, if it's on the wording, on glitches, anything. You can do it on Discord, on the contact us page, or reach out to me directly on Slack or anything. And we welcome our feedback along the way. Like, we know topics such as indigenous data sovereignty and engagement are constantly evolving, and we want to keep following that. But we may miss some things, so we always welcome feedback. And also, as I said, stay tuned for our next set of conservation challenges that will be posted very soon. And we have a shared talk during Biodiversity Genomics conference. So I have a shared talk with Gustavo Bravo. It is during the Indigenous Data Sovereignty in Biodiversity Genomics session on November 1st. So I invite you to attend all of BG24, of course, but especially this session, because I think it's going to be very, very interesting. And I think that's about it. Yeah, thanks. I'm the lucky one to present and launch that to all of you, but that's been the work of many, many people in the team and our collaborators in Bermuda and our collaborators in the next set of challenges. We all came together to develop the platform and to test the model. So we're very happy with that. And yeah, thank you for attending, maybe registering. Yeah, now it's the end.
Thank you very much. It is very cool. Um, for the subsections on the challenge page, will there be a place for even like a PDF just to describe what you expect of the genome? Just so then people have a better understanding signing up for it, like expected genome size and the expected amount of data that you're looking to get and that kind of thing.
So, yes, so let me share my screen again. Hopefully you will find something that we haven't put on the website or that it's not easy to find, and we'll improve it so that it gets easier. I can here. So for each species, sorry, my connection is a bit slow. There is a species information tab.
Oh, okay.
Or they should be, but you're right that where there is the genome size, there is information. We haven't made it public. Is it just for us to keep track? That's a good point. Okay, yes, there should be. So we have that information because I checked before, like if it's already in EVP, like if it's already, if there is a reference genome already, if another project's working on it and all of that. So we have that information. Maybe it's not, we don't want to overwhelm people with information either. So it's about finding the right balance of what's displayed and what's not.
That's fair. Yeah.
But for example, you were asking about the coverage. So for the sequencing, we do say in the phase data that we expect a coverage of 10x per sample. We also understand that sometimes you have expectation, and if the sample is not of good quality or if the extraction failed, you cannot get those 10x. And so for the bioinformatic analysis, kind of, we could say like you will have 10x of long reads, but it also depends on what the previous collaborator is able to do. So do you think a PDF would be more helpful, like something people could download and look at? More engaging for people?
I mean, maybe, but I know a lot of people that just want the hard facts straight in their face as soon as they open the tab. So it really depends on the sort of person that you're getting to look at it, doesn't it?
Because we could do it quite easily. We could
Yeah.
create a PDF and like have people would have the choice whether it's the things displayed here or you have download PDF and you can have the same information, maybe displayed slightly differently. Yeah, that's a great idea.
Yeah.
I'll write it down. That's a new task for us. Thank you, Damon.
You're welcome.
No, but exactly, we're welcoming this type of feedback. Different people work differently, and we really want a model and a platform that works for everyone. So let us know. And
yeah, legitimately, this is an incredibly cool project, and I will be applying for at least one of them. So
cool, great. Yeah, we're excited. So for this, the Bermuda project, the sample extractions and sample processing and the sequencing will happen in Bermuda because they have the platform. But we are looking for bioinformaticians for both challenges, the reference genome and annotation and the identifying species-specific region to run the genetic test. So yes, please apply. We will also, you can follow us on social media. We'll have specific targeted posts when we are looking for example, bioinformaticians or platform with exactly the information you were saying, because we understand some people may not look at the platform, but are on social media a lot. Some people are not on social media, so the information will also be available on the platform. We're trying to reach out as much as possible.
Cool. And with will there be priority? Well, I imagine there would be, but priority given to people that are essentially the closer to the area of interest will be given priority. So
Not necessarily. Like for example, I mean, people in Bermuda, technically the US are closer than Canada or than Europe, but as long as it's outside of Bermuda, I don't think they will have a preference whether it happens in
okay.
one country or in another. So I don't think geographically there would be the case. It's more if there is a wish from the local community to have knowledge transfer. They may want people they know they can reach out to or have, I don't know, weekly or monthly meetings with, just to learn more about like what's that type of processing, what's happening. So it's also, I think, about more of a fit between people more than geographic or regional fit. But we will, if there is local capacity, we will definitely try to support that. But as long as it has to be outside, I don't think geography is going to matter a lot. There may be some countries who have embargo working with other countries. That those are the kind of thing we ask, and we've so far have like no everyone can apply for challenges, but we ask in case there are things we may not be aware of.
Okay, cool.
Yeah.
Awesome. Right. Does anyone
Well,
have
everyone for
any
attending.
questions?
I want to thank everyone, saying that it's a very exciting project. We're very happy. I think so. And yeah, share, like the talk will be available on YouTube, share it. I don't know if you work in private companies or in academia, like share it with your colleagues. Please, we really need to grow our community if we want to convince funders that it's a model that can work.
Cool. Right. Well, if there's no more questions, should we let Celine get on with the day? Right. Well, thank you all very much for coming along. It's been a fantastic session. Thank you, Celine, for giving up your time to run the session. And I will see you all in the next one. Thank you.
Thanks, everyone. Bye.